an inch.
Okay.
An inch and a share. I think it's this one. Yeah.
Okay, I'm gonna get plugged in. Oh, there's also one like mounted here, it looks like.
Yeah. Yep. Yep.
So. And even though I'm fully charged, I'm gonna plug in just in case.
Do you want a water? Or you
I brought a water bottle. Yep.
I have more if you need it.
Hello.
There she is.
I'll silence it now. Don't need that going off. Okay. So, yeah, we'll go in the HDMI there. Ta-da! Okay.
Ta-da! Woo-hoo!
Does your presentation has any sound on it?
No sound, just a
No
lot of
sound.
little animations.
Okay.
I'm gonna be yeah, I'll use my little
Pen,
it's the side
clicker.
clicker. So we'll just run through it here. There's for Robert, he wanted me to bring it on a USB, a final, just in case.
Okay. I have.
So,
Maybe they will be able to give it the video person.
okay. Good, I should probably turn this on.
Is that for Sharlin? It's a USB thing that Robert ordered. Can you get that to him? Oh, it's right. Okay.
If he's, there's a bunch of stuff on here. There's a bunch of stuff on here, so I want it back.
Yeah.
But if he just sorts by the date, it would say like half hour.
Okay. Sounds
That's
good.
fine.
Okay, that is.
Yeah.
Thank you.
It's a clock, yeah.
With the lapel mic on it.
Yeah, let's, should we test that too?
Yep.
Okay. Whoa, let's turn it off for me. What do you think? Pocket?
Gosh, I think I'm gonna move over here.
Hold on a second. Where do you want it? Like I don't.
Where do you want this round about it?
Any way
Anywhere.
you're comfortable with it.
Can you,
Or maybe do you want it even, actually it probably be
can-
better here, now that I think about teaching.
We can turn it on and off.
This thing. Do this in C121 all the time, so. Maybe it's better if I,
You wanna put it
my waistband, hide it a little bit.
where?
Check. That seems super loud.
I can lower it down.
Okay.
I think that's really good though.
I guess you'll monitor it anyway, right? So
Yeah.
if I turn on my teacher voice, you'll be, oh yeah, you were here at the practice.
Yeah.
Yeah, so if I turn on my teacher voice, then you can always bring it down a little bit.
Sure. There will be like more people in here, so.
Yeah.
But yeah, we'll adjust it.
Yeah.
I will like suggest to like keep it off when you're not using the little mic.
Okay. So right now it's okay though, right?
Yeah.
All right, turn it off. There it is.
Ta-da. Oh, there we go, it's working.
Working good.
Looks really good.
I've added a few things. It's running like 32 minutes, 33.
Okay.
Is that okay? I know you said
Yeah.
30. I'll probably talk faster now than I do in my office. So.
Yeah, it'll be just great.
Yeah.
So I will cut off questions probably around, let's see. I don't know, let's see. Should
Alex?
this, the whole thing take an hour, are we thinking?
I think so. Right?
Yeah, I think so too.
And Allison's on her way. She just texted me. She can bring her computer, so
Awesome.
if people want to send comments in through the
Yeah,
YouTube chat.
yeah, exactly.
Now look okay? Like,
You look very
okay.
nice.
I told you I don't get dressed up very often, so
No, it's pretty impressive.
I'll leave that
See, now
there.
I was gonna sit here, but it's really not very good, is it?
You might get cut off by this, right?
Yeah.
I mean, I'm gonna walk around.
Yeah, you're gonna walk around. Okay,
It's
so.
kind of my thing, so
that makes-
being able to interact with the slides a little bit.
Yep,
And, at the end, I'll do like the knee slide into this camera.
yeah, yeah, I think that'd be really good, really good. And let's see if we get President Mitchell do it too.
Yeah, we get together. Well, I mean, after that win on Saturday night, like,
Right.
you kind of wanted to.
Exactly.
Wow.
I didn't see it, but I heard
Oh.
about it, so you were there?
I wasn't at the game, I watched it on they stream it now
Yeah.
on the local TV.
Nice.
So, okay. Oh, thank you.
Perfect.
Yeah, I do a big vegetable garden every year,
Oh,
and I take over half my kitchen and turn it into like seed starting area.
Oh.
So I did that Saturday, like during the game, like, I don't know, probably started 500 plants.
Oh my
Maybe not that
gosh.
much, but a lot.
That's impressive,
Maybe 300.
David.
Yeah. So we do a big plot. Feed ourselves all summer.
Nice. Do you live in town?
No, we live kind of outside St. Clair.
Okay.
9
Oh, yeah,
acre
yeah,
little
yeah.
farm.
Oh,
Yeah.
very nice. Yeah. I live on 16 acres in Rotana.
Okay.
Half acre spoon-fed pond.
Beautiful.
Oh, it really is, yeah. But it was my parents, and I bought it from them a couple of years ago, renovated upstairs. I lived downstairs for years so
Okay.
that they could stay there. And then this year I actually sold my house that I had been renting and then renovated this one, and then my mom just recently moved into assisted living.
Okay.
So it's weird, it's pretty lonely out there. It's like,
Yeah.
it used to be crazy busy, and now it's like silent.
So some of my best friends just showed up. Yeah, come down from the cities.
Heaven.
And yeah, we're on a big wetland in St. Clair.
Oh, awesome, that is great. Yes.
Oh, yes. Two childhood friends of mine. That's right.
You mean it's too childish friends?
Oh, yeah.
Yeah, that too.
Are you gonna comment on
Dapper Dick.
With my gigantic screen behind me.
Yeah. Do you have a link? Put it online there? Oh, there it is. I'm gonna take another one. Do that again.
You can, Bob and Nancy will be on YouTube.
Yeah.
Which camera is that?
What's that?
Which camera
I don't
goes?
have it.
All of them.
Thank you.
All right. Which one do I move?
Oh, I don't know, they look like, there's a guy for production, producing.
Stop.
I know I was joking earlier that I'm not gonna go right into this one.
Yeah, you should.
Grab the collar. That will shake.
Thanks for moderating the channel.
Yeah, of course.
Is that what it looks like right now?
Yeah. Yeah, just make us big.
Channel live.
Ain't that big.
Well, not as big as yours are, mine is.
There were no responses. So
Yeah, just type 5 words in and it'll take care of it.
Hello, everybody. Hi, everyone. We're going to get started tonight. First, let me just welcome you here, those of you who are in the room. Provost Hood, good to see you, and President Inch is here. We have a couple of deans from the college, pre and post. How about current and post? Good to see you all here. Thank you. So this is one of our most special events in terms of the research, scholarly, and creative works. And so it's a really big deal tonight. My name is Terry Wallace, and I serve as the Interim Associate Vice President for Research and the Dean of University Extended Campus. My job tonight is really just to walk through what's already in your program. And then I get the opportunity to introduce our president as well. So after hearing from Dr. Sharlin today, we will take some questions, and you'll see that it's different this time than it has been in the past. So we have a couple of microphones in the audience here, and then Dr. Land, raise your hand. She is taking any questions that will, oh, other people are waving at me. Other questions that will happen online. So that'll be really great and very much more inclusive than we were last time. So when you ask questions though, some of you are gonna wanna just yell it out in the room, and that won't work. In order for everyone online to be able to hear you, make sure and come up here and use the microphone, okay? So after question and answers, then our President Inch is going to return up here. He's going to congratulate Dr. Sharlin on his award and then announce the 2024 award, which is really exciting. So after that, then there will be a reception outside, right outside here. Those of you online, I'm sorry. We don't have anything to send to you. But please join us out there for a little bit of networking and congratulating our recipient. Before I exit, I really want to take a minute to thank the selection committee members this year and last year. Also, President Office team, Robert and his team. These folks have been here now for a couple of hours getting ready with the video and the microphones and testing everything, and so we're really grateful to them. And then especially my colleague, Julie Jorg, who has been keeping us all together and moving all of us forward really successfully. So with that, it's my sincere pleasure to introduce and invite President Inch, our 13th president, to the stage.
Well, thank you, Dr. Wallace. That was very kind. Good evening, everybody. Those of you who could be here in person and those of you who are online, I think this will be a fascinating evening with a lot of opportunity to learn and grow. This lectureship is one of those very special things that universities do, is celebrate the very best kinds of research that we do, to think about the very insights that we're able to discover on our campus and the conversations among colleagues and students, faculty and staff that allow us to dive deeper into subjects and ideas and explore things that have never been explored before. And that's why I'm very, very happy to welcome you all to a special evening that dives deeply into subjects that many of you probably don't know much about. I certainly do not. And it's our 49th annual Douglas R. Moore Faculty Research Lecture. And it highlights the incredible work being done on our campus. And I can't say enough about this. This is one lecture today that is the best of the best. But this is one lecture of many things that go on our campus, in our classrooms, at conferences where our faculty display what it is they've been able to discover on our campus or be able to apply with our students in such exciting ways. The person who is selected this year for the lectureship is Dr. David Sharlin, professor in the Department of Biological Sciences. His presentation, Thyroid Hormone Disrupting Chemicals in Nervous System Development, a Growing Cause for Concern, centers around the understanding the sensitivity of the developing nervous system to perturbations in thyroid hormone signaling. I think this will be fascinating. It's certainly an area that we all grapple in about health and about how it is we grow, develop, and how we stay healthy. And so please join me in welcoming Dr. David Sharlin to the stage.
we on here? I think so. Thank you for the introduction, President Inch. I do want to start off by just really saying how humbled I am to be selected as the Douglas R. Moore Lectureship recipient. To join that list of faculty, you know, engineers, other scientists, artists, poets that have come before me, it's truly an honor, right? And so it also kind of validates the work that my students, some of them sitting over here, do, and that I do, and that it's important, and that it's seen as something that is worthy of such an award. So, as President Inch indicated, I'm going to talk today about environmental chemicals, thyroid hormone action and development, and the idea that maybe this is a concern. We're going to try to link these two together. I'm going to talk about some unknowns. I'm going to talk about some uncertainties. I'm even going to show data that wasn't predicted based on how we think things work. So before I get going, I really need to start off by acknowledging most of the folks that do the work. It isn't always me. I've worked with a number of graduate students and undergraduate students in my 10.5 years here now. And some of their work I will discuss tonight. So in the names behind me that you see are bolded, those are students who are either currently in my lab or have recently departed that have contributed to some of the data I'm going to share with you. I also need to acknowledge certainly our collaborators from around the world who have not only provided, you know, platforms for discussion on the things that we do, but they have provided resources, including mice. We have Swedish mice in the lab, German mice in the lab, have come across the Atlantic to do some of the work that we do. And I'm not going to talk about that work, but these collaborators are essential for some of our experiments. And lastly, and also as important, are folks that who have paid for all of this, right? The work isn't free, it costs money, although most of the students work for free, thank you. Actually, they pay because they take credits. So I've been fortunate to receive faculty funding through the university's sponsored, research and sponsored programs. My students have received funding from the University Foundation, from the graduate school, from the Undergraduate Research Center. The American Thyroid Association, which is the largest collection of dorky thyroidologists like myself, has funded our work as well. So they definitely need to be thanked as we go forward. All right, so let's get started. So thyroid hormone is kind of best known for its role in metabolism, right? Now, this idea has to do, the idea that thyroid hormone regulates metabolism has to do with the fact that individuals that suffer from hypothyroidism, so this is a condition where they don't produce enough thyroid hormone that's circulating through their blood, have kind of metabolic dysfunction. They gain weight, right? They're cold, they're lethargic, right? And this is because when you don't have sufficient thyroid hormone circulating, your metabolism at the cellular level is depressed. The complete opposite is true in individuals that have excess thyroid hormone, right? They're insomniacs, they can't sleep, they're hot, they're losing weight, right? And this is because their metabolism is cranking, right? Now, what's less appreciated and where we come in at this is that thyroid hormone is also essential for development. Both the maternal thyroid gland, which actually increases its output of thyroid hormone during pregnancy, as well as the fetal thyroid gland that starts to develop during the second trimester and start kicking out its own thyroid hormone, are important in regulating developmental events in the embryo, in the fetus. And probably one of the best examples I can give you for kind of the importance of thyroid hormone in development is the fact that every child born in this country after 1976 is screened for thyroid function at birth. If there's someone in this room that's had a child, you might remember this, right? They take, they prick the heel, they take a little bit of drop of blood, they put it on a piece of filter paper, and in that filter paper, they measure something called thyroid stimulating hormone. And this hormone, TSH, is a really good indicator of how this thyroid axis that regulates our hormone in kind of a normal range tells us how it's functioning. And if we can identify this child and treat them early, it actually reduces a lot of the deficits that I'll get into that will come later. So I'm going to take us back 130 years. Okay, so this is a study from Sir William Osler, who was a physician, 1897, right? I certainly wasn't born. Now, in this picture, this is a young boy. He's 4 years old. He has been diagnosed at the time with something called sporadic cretinism. Today, this would be called untreated congenital hypothyroidism, being born with an improperly functioning thyroid gland. And unfortunately, this child, 4 years old, short stature, so he's not growing appropriately in his height. He actually can't even stand. This cloth you see here, there's someone behind him actually holding him up. He has metabolic dysfunction, which can be seen in the extended abdomen. He has an unaware gaze, unaware gaze in his look, and he has such poor motor control that he can't even hold his tongue in his mouth. This is the same boy treated with thyroid gland extract from a sheep for 4 months. Right? It doesn't take a lot to realize there's marked improvement. Okay? He can now actually stand up on his own. Somatic growth has taken off. That like unaware gaze is gone. His metabolic metabolic issues are recovering. However, what would not be recovered is the fact that this child would still have severe neurologic deficits, low IQ. He would also have auditory impairments. And the work I'm going to talk about today leans into the auditory side of things. The lab also works on some of the brain development issues, but I'm going to talk about our work in the auditory system. So today, this boy would have actually been identified by neonatal screening and treated. And although he would have been identified and treated, epidemiological studies still indicate that there are residual deficits, including hearing deficits. Okay? All right, so to really understand what's happening, we're going to dive into a little bit of molecular biology and look at how the hormone actually works. So thyroid hormone is secreted by your thyroid gland, which if you can find your Adam's apple, it's just below that. So it's right across your windpipe. And it secretes 2 hormones. One is called thyroxine. I'm going to call it T4 for short. This is inactive, actually. And the majority of the hormone that the gland secretes is this inactive form. And it's characterized by, I just circled them here, by 4 iodine molecules that have been conjugated onto this molecule. And the fact that thyroid hormones are iodinated, they have iodine, is the exact reason why we have iodized salt. Especially in parts of the country, like where we are now, which once was called the goiter belt, which was an enlarged thyroid because we couldn't make enough thyroid hormone. There's no iodine in our food here. We don't eat things out of the ocean. We iodize salt. This fixed the problem. Now, the second hormone it's secreting is something called, big fancy word, triiodothyronine, and they call it T3. So it's actually the active form, and it's characterized by 3 iodines. So to go from the inactive, 4 iodines, to active, 3 iodines, the only difference is the removal of a single iodine from this molecule. These hormones are secreted from the gland, they enter our blood, and they're circulating in the blood of all of us in this room. Ultimately, the hormone moves out of your blood and into cells and tissues, okay? And so once it gets into the tissues, they have this really unique capacity to actually further regulate the availability of the hormone to do something. We'll talk about what that something is here in a second. But we have these enzymes, and I'm introducing these because they're going to come back later when we talk about kind of maybe the system is more sensitive than we think it is. And so the first enzyme I'm going to introduce is something called DIO2. It's a type 2 deiodinase. Actually, what it does is it converts this prohormone, T4, inactive, to the active form, T3. This happens in the tissue. It's creating its own supply of the active hormone. This hormone then actually, T3, enters the nucleus of a cell, binds a receptor. That receptor has a nice name. It's called thyroid hormone receptor. And it's a transcription factor. And that's just a fancy word for saying that it's a protein that has the ability to turn genes on and off. Okay, so thyroid hormone, ultimately what it does is it regulates gene expression. The tissues also have an enzyme that can turn off the hormone. They take that active T3, they turn it into a metabolite that doesn't work at the receptor. Okay? So when we think about slides before this that I just went through about importance in pregnancy, importance in neurodevelopment, ultimately, it's the misregulation of DNA, gene expression, in developmental hyperthyroidism that's the underlying cause of these observed defects. And this is what my lab ultimately is interested in. How does the hormone work? What are the genes? And then how do those genes translate into developmental processes like making the ear work, making sure our brain works appropriately? All right, so we're also going to talk about environmental chemicals. So I'm using a definition, endocrine disrupting chemicals, I'm using a definition from the Endocrine Society's statement on EDCs. And they say an endocrine disrupting chemical is defined as an exogenous chemical or mixture of chemicals that interfere with any aspect of hormone action. So let's layer that on top of thyroid hormone. So if we want to know what a thyroid disrupting chemical is, it's going to be an exogenous chemical or a mixture of chemicals that can interfere with any aspect of thyroid hormone action. We just saw on that kind of complicated slide, right? So a chemical could block synthesis. A chemical could block secretion. A chemical could block its ability to have a half-life in our serum. A chemical could block its ability to move from blood into tissue. A chemical could block the ability of the deiodinase enzymes to work. A chemical could block the ability of the receptor to work, right? We don't know. Well, I'll share a little bit on what we do know, but it's kind of scary to think about that these chemicals might be able to perturb this system. So what are chemicals we're talking about? I'm going to put 3 up here. This is a very short list. And the first one I'm going to introduce is something called triclosan. Has anybody heard of this chemical before? Yeah? So triclosan is an antimicrobial agent. Up until 2016, it was loaded in hand soaps. All Dial soaps had it. Was banned in 2016 when it was realized that washing your hands with soap with triclosan was no more effective than just washing your hands with soap without triclosan, right? But then triclosan ends up in our sewage water and goes into our lakes and our rivers and goes out in the environment, right? Structurally, so I also added that thyroxine molecule, the thyroid hormone here. Structurally, it's very similar, right? It's got these rings here. It's even got this ether link in the middle. It's got these large chlorine molecules, which are kind of similar to iodine. Right? So structurally, very similar chemical. The one in the middle here, polychlorinated biphenyls. This is like the poster child of endocrine disruptors. I worked on this as a graduate student. You might have heard of these chemicals before. They're ubiquitously in the environment. Again, structurally similar to thyroid hormone. They were used as like heat transfer liquids. There were billions of pounds of these things made until 1979 when the production was banned in this country. 20% of that has been released into the environment, and 10% of what produced is still in use today, right? I wouldn't be surprised if there are transformers on our campus that contain PCBs. Lastly, and I added this one here, not that we work on this one, but this is the one that's in the news right now, right? These are these forever chemicals, right? These are your PFAS, these polyfluoroalkyl substances. These are the things, your Scotch guards that like our Minnesota company 3M made. DuPont's Teflon, right? There's other non-stick coatings, waterproof coatings. These are these PFAS. These are called forever chemicals. You look at this thing, it is super stable. They get in the environment, they're not going anywhere. And probably the largest exposure right now is through drinking water, right? And just last month, EPA set drinking water standards that probably 20,000 wells across this country won't meet. PFAS contamination. All of these, as well as most of the other chemicals that we know that disrupt thyroid hormone, do so by reducing circulating levels of thyroid hormone in the blood. That's a regulator's like first line of kind of detection here, like does it reduce hormones? And you can do these studies in humans. You can look at what's in their blood. You can associate them with thyroid hormone levels, TH levels, and we can also clearly do these things, treat mice and rats with these chemicals and watch what happens. Okay, in my lab, as the front of our program indicates, we work in mouse models. So now, I don't want to scare you all here, but the heading probably tells you something you might have already known, and that's the unfortunate truth that every child born in this country is contaminated with synthetic chemicals, right? And I've been using this benchmark study now for, I don't know, it's almost done 20 years ago. It's been repeated since, probably been 10 additional since this time. But what they did is they randomly chose newborn baby cord blood samples across the country from early fall 2004. And in that cord blood, in that sample, these scientists looked for 413 chemicals, and they found 287 across these 10 samples, about 70% of the chemicals. And they were, could fit into 9 diverse chemical families. Now, this is kind of a summary of all the chemicals that the families that they saw, and there's a lot of information on here, but I'm going to highlight 2. So the first is these perfluorinated chemicals. Your PFOAs, your PFAS, your Scotchgards, your Teflons, your coatings, right? Just told you these are known to reduce circulating levels of thyroid hormone, right? 10 of 10 newborns tested had these perfluorinated chemicals in their cord blood. The other one at the bottom here, which isn't surprising actually, is the polychlorinated biphenyls, right? Also 10 of 10. My guess is that every single one of us in this room have both of these chemicals circulating in our blood right now. And lots of questions, where did we get them from? We can answer those later, right? All right, so let's bring these two sides together, this thyroid environment concern, right? So I just spent first, I don't know how many minutes telling you about thyroid hormone. It's essential in development, right? And now I just told you there's all these chemicals in the environment that can maybe disrupt thyroid hormone. And so, there's questions whether or not these environmental factors, these chemicals, can interfere with maternal or fetal thyroid function and disrupt development, right? These are big questions. We need to be looking and thinking about these things. However, to understand if these chemicals can disrupt thyroid hormone action, we also need to understand how they're working, like what are they doing? And even though 130 years ago, it was demonstrated very clearly that something from this thyroid gland extract could do remarkable things in a child with congenital hypothyroidism, we're still a long way from understanding what the hormone is actually doing. What are the genes? How do those genes translate to mediating developmental events so we have the ability to think and hear and speak and do all of these things? So my lab has been kind of focusing on 2 questions that are related to this more recently, and this is some of the data I'm going to share with you. So the first is it's not clear to what extent adaptive mechanisms, I'll come back to this word, provide compensation and can protect developing tissues. Remember these deiodinase enzymes we talked about? Can activate, inactivate. So the idea in the field is that they respond in ways to try to normalize what's going on in the tissue. So if thyroid hormone levels fall in the serum, you can turn on this D2 and create a bunch more locally, right? And maybe at the same time, take off the brake, right? The other question that we're looking at is, to believe it or not, and I'm guilty of this too, this is why we're doing this work, is it's not clear to what extent you actually need to reduce thyroid hormone levels to see changes in gene expression. You know, when we were doing this, a decade ago and more recently, like, we would nail the animals, right? We wanted to see changes, right? But it never gave us the information on how sensitive things are. And so we need to circle back now and kind of ask that question. And so to do this in the lab, we're actually using mouse auditory system development as our model, okay? And we're doing this because we know it's important in hearing development in humans. We know it in mice too. And so what we can do is we can use, right now, we're mainly using kind of prototypical thyroid gland inhibitors to reduce circulating levels, right? We can treat these pregnant moms, we can look in their offspring. Oops. And so really what we're trying to do is tie exposure to changes in the tissue to functional outcomes. Because believe it or not, we can test hearing in a mouse. We've adapted the same system that they use to test hearing in a neonate. We adapted that to work in mice, and we can detect changes in the brainstem. So our goal is to link all this together. So before I get into some data here, just a little bit about the auditory system. So we hear because your ear collects sound waves, goes down that external auditory canal where you stick a Q-tip. You know, where actually, has anyone ever read the label on a box of Q-tips? It says, do not stick in ear. We do, right? Anyway, so here's where your Q-tip goes. To the end of that canal is your eardrum. It vibrates. I think we all know that. On the other side of that eardrum are those 3 smallest bones in the human body. These are your ossicles, and they vibrate in response to this eardrum. And the last one, the most medial of these bones, sits on this spiral organ that's embedded in the bone behind your ear called the cochlea. And inside the cochlea, this is the hearing organ. This is where sound is detected. Right in here, if I blow this up a little bit, you'll see that there are these cells that are called hair cells. These actually have this membrane that sits on top of them, and they get activated in a frequency-dependent manner by bending the little hairs. When the hairs get bent, they activate, and then they send a signal to a neuron that actually then goes through your brain. It goes to your brainstem, goes to your midbrain, and finally it gets up to your auditory cortex, and all these electrical signals are decoded so you can make sense of the sounds, okay? And then lastly, I just need to point out, so this is a mouse. This is a human illustration. This is a mouse, looks just the same. 4 rows of hair cell, this membrane that sits on top. And the reason why I put this up is because we and others have done a good job of trying to understand how this develops in a mouse. So what I'm about to show you that's happening during the first 2 weeks of life in a mouse happens during the 2nd trimester in utero in a human. And so what you can see is that there are those hair cells. They're in, they're highlighted by these arrows. Just going to leave the little thing there, sorry. And so if you notice, there's this population of cells. I'll bring them up in green, right? It's called the greater epithelial ridge. These cells, their importance is actually they're secreting the proteins that are going to make the membrane here that sits on top. So if you follow kind of the green in the arrow, eventually these cells disappear. They leave behind this membrane. Also, there's an opening that occurs between this inner hair cell and these other hair cells. And we can use these endpoints as distinct developmental markers of what's happening in the cochlea, right? And moreover, we know and have seen that thyroid hormone regulates this timing, right? So here are those cells. Here's a hypothyroid animal. Here's that membrane. Here's the normal animal. You can see those cells are still present. They're still making all of this membrane proteins. And you get this grossly overgrown tectoral membrane. Interestingly, if you give a mouse excess thyroid hormone, you speed up the whole process, right? These cells disappear sooner. That tunnel opens up sooner. Hair cells are still all there, right? It's all this timing that's happening in this development that's being regulated by thyroid hormone. The kicker is both of these animals, hypothyroid and hyperthyroid, would be deaf, right? So we have these really nice endpoints now to be able to start asking questions about sensitivity, about compensation, and then ultimately about environmental chemicals. So the first thing we've been working on, is trying to understand this compensatory, I don't know how to turn that off, sorry. this compensatory mechanism. So on the left side here, this is embryonic development. On the right side, this is postnatal development. And the graph is depicting the levels of these two enzymes. The restricting enzyme DIO3 and the activating enzyme DIO2. This is what we know, right? And so it looks like early on you're trying to restrict hormone signaling. Later you want to amplify it. If these are working as compensatory mechanisms and adapting to low thyroid hormone, if the DIO3 is a brake, you take off the brake, right? Reduce it. At the same time, you would turn on the gas. Move forward this activating enzyme. This is adaptation. If this works, then you can help protect our developing tissues. So I had a graduate student, Maria, who asked this question, right? She did the experiment. Took pregnant mouse, treated them with our well-known and favorite thyroid gland inhibitor, looked at the offspring by investigating the deionases. What did she see? Well, I should say, so Maria now, PhD student at Cincinnati Children's. She's a third-year PhD student. She's in a developmental and molecular biology program. So what did she see? So the first thing she did is actually did a technique to visualize the RNA. This is that hypothyroid animal. You can tell it's lower. Purple means the enzyme is being expressed. It's lower, okay? She also quantified it using PCR, which is the same kind of methodology that one uses to like screen for COVID, right? She used PCR to quantify the amount of this deionase. And if you look closely, that first window where things are really changing, you can see that not only in some cases it's significantly lower, it's definitely not amplified, right? So this is actually quite counterintuitive to the dogma of our field that says these things are adapting and protecting our tissues, right? She also looked at the DIO3. A little more complicated here, but the take home on this is it didn't drop. At one time it did, but otherwise it was same to these control animals. So our interpretation, Maria's interpretation of these results, is that they're not responding in a mechanism that would be consistent with the idea that these deionase enzymes are protecting the tissue, at least not in developmental situations, right? And this kind of idea that they work as adaptive mechanisms to protect tissues comes from studies in adult animals. Developing animal is not an adult. I think we all would agree with that. And so it looks like that maybe the programs that are regulating these things developmentally are dominant over their actually ability to respond in ways to protect. And so future work is going to look very closely at the genome level of how DNA is actually regulated to see if that's the case. Maybe they can't respond because it's silenced. All right, so the other ongoing question that we're addressing is done currently by a number of undergraduates in the lab. So Jessica recently graduated. She's now a DO student in Des Moines. Ainsley is a junior here, sitting over here. She's a biochemistry student. James, who's also over here in the lab, is a sophomore biomedical sciences. And we've been doing this experiment where we're trying to create graded levels of loss in thyroid hormone circulating, right? So rather than just giving them one big dose and trying to see what happens, let's incrementally decrease thyroid hormone and look for that tipping point, right? So if you do a 10% reduction, 20, and so on, at what point do you start to see things change, okay? And so they set this up, and the gene that we're starting, that we've looked at one so far, is something called tectorin beta. This is the protein from this gene, is what makes up the majority of that membrane that sits on the hair cells. The only place it is expressed in the animal is in the cochlea. And our previous work indicated that it falls in hypothyroidism, and if you make the animal hyperthyroidism, it's overexpressed. So it seems to be regulated by thyroid hormone. We want to use this as an endpoint. So they did the study. What did they see? This is their serum data, right? And I love this because it's a very mild to moderate decrease. It's not all the way down here at the bottom like where we've usually been doing things, right? So now we can actually ask this question, what happens to gene expression in the tissue? What do you guys think happened? Okay, no guesses. That's all right. Not at all what we expected. It went the opposite direction. Based on our previous work, where we're looking at severely hypothyroid animals, it decreases. We actually saw an increase. And I really wish we were able to do the histology in these animals, because this effect says that they're hyperthyroid, which was not what you would predict based on the serum, right? So, you know, I didn't purposely showing you this because it's complicated, and there's a lot we don't know, and systems aren't responding as we would predict. I think it just adds to the fact that we want to try to protect ourselves from these chemicals in the environment. We still need to understand how the hormone is working, how sensitive the tissues are, so we can actually do, you know, regulation that's appropriate. Now, I do want to bring it back to endocrine disruptors. So we think that we can use the work we're developing with the auditory system to very specifically look at these thyroid disrupting chemicals. And we have done some work with triclosan. We started these a few years ago with the help of a FRG, a faculty research grant from the university. And so we treated pregnant moms with triclosan antimicrobial. This is a 100 milligram per kilogram per day dose that we gave the pregnant moms. And this is about 3 milligrams a day. It's not a lot, but it is more than a normal person would probably ingest in a day. And we have our control animals, which are MMI. This is the drug that we've been using. And so you can see in these 7-day-old animals, it actually reduced circulating levels of thyroid hormone. 3 milligrams a day, that's a couple grains of salt, right? It was able to reduce circulating levels of hormones. However, mice that were allowed to go out to 15 days, there was no difference, right? And I think what this speaks to is that the way these chemicals can be transferred is going to differ. So the P7 animal, probably much of what it has on board for triclosan came across the placenta, where much of what that mouse has at 15 days old probably came from the milk, right? And so the route of transmission is probably going to have very specific, maybe effects on kind of the kinetics that happens. We've done a little bit of histology, and so this is that 7-day-old mouse here, and these are those histological endpoints. And we do think that we need to do quantification on this. We need more numbers, but that tunnel is not opened here, but it's opening in the euthyroid animal. Those cells, there's more of them gone than there are here, right? So we have a long ways to go, so stay tuned for updates on this. So to conclude, I just want to come back to the overall overarching kind of work that we're doing. So the lab first wants to understand what is the hormone doing? How sensitive are the systems that the hormone's working in? What's the tipping point, right? How well do our tissues adapt? Can they compensate? Then we want to take this information and apply it to testing environmental chemicals that we're exposed to on daily events. And the idea is to link these together with the ultimate goal is to have some sort of risk, provide information for risk assessment so we can really inform and protect human health. Because that's the main reason why we're doing this, is to make sure that our society stays healthy. And so with that, I thank you all for your attendance, both here and online, and I'm happy to answer any questions, and I'll leave you with a beautiful picture of mouse hair cells.
Questions? Anybody want to come up and ask a question? Anybody online? Dr. Land? Okay.
Someone's got to break the
Could
ice.
you? Here, I'll bring it to you.
Lyric.
Do you foresee there being treatment for people in our generation, and I guess before now, being treated for these like PFAS chemicals that you said affected us. So if we're treating, or later on it becomes regular treatment for perhaps people who are pregnant, do you see then people who have been exposed to it being treated post-development?
So I think, you know, a lot of the effects are permanent, right? So any treatment that would likely need to occur are going to be, you know, more assistive, trying to help an individual, you know, accommodate whatever they're being afflicted with, right? There's no really turning back that developmental clock. If the ear, which I talked about, you know, now you have permanent hearing loss, you know, being able to restore hearing, it probably doesn't have to do with the PFAS. That's going to be a whole different side of research to try to fix hearing, right? Is that answering your question?
Yeah, that answered my question. So
you know, the idea with the good news and bad news is that, like, the original formulations of Teflon and Scotch Guard are basically banned. But these companies, they modify them. You know, you still buy Scotch Guard, but its formulation is different now. So now it's rather than it being this alkyl carbon chain, they're using like urethane, fluorinated urethane, right? Like, still not tested, still don't know what's going on, right? And some chemicals, right, have benefits, and you know, so it's a balance on what gets used, I think. I love it. These are my students. They're asking questions.
We've talked a lot about IVF. Do you foresee there being types of things with like the genes in the embryos, or being able to test right away once the egg is fertilized for these type of hypothyroidism or hyperthyroidism?
Yeah. So there are certain types of genetics that lead to like thyroid dysgenesis. So like not having a thyroid being born. There's a couple of well-known genes that cause that. So certainly if you have those kind of traits in your family, you could certainly do pre-implantation screening to make sure you're picking embryos in a way that allowed you to use one that doesn't contain the mutation. But much of the hypothyroidism and congenital hypothyroidism, which its prevalence has doubled in the past 20 years, we don't know the cause, right? It's de novo. It happened for a lot of reasons. That get you? An online question.
Hi, Alec Online asks, do you think the timing of exposure to different chemicals may change the severity of the effects or mode of entry of the chemicals?
Oh, that's a very good question. So, does the timing of exposure and mode of entry? So if we talk about the auditory system specifically, it's a really good question, Alex. So I didn't get into it, but the cochlea actually develops from the base of the cochlea to the apex of the cochlea. And it's also frequency dependent. So at the base, basically where that stapes, that one bone is sitting, that detects high frequency, which is like, I joke in my anatomy class when I talk about high frequency, it's like preteen at a One Direction concert. And so very high pitched sound. The other end of the cochlea is the bassy noises, right? The low frequency sounds. And this develops, the cochlea develops from base to apex. And so during those 2 weeks postnatally, if you disrupt development early, you can disrupt more high frequency sound detection. If you do it later, it'll be more low frequency development. And same with like the mode, like I guess it gets at the triclosan results we have, placental versus postnatal. You know, there's a lot of development that happens in utero that would be different than the effects that could happen postnatally. So depending on what time you just kind of perturb the system. So yes, simple answer.
Simple. Other questions? Yeah.
Have you been to a One Direction concert?
No.
Actually, my real question is, are you aware of any research being done to eliminate or remove these chemicals from the environment, the PCBs and the PFAS?
Yeah, so good question. So, I mean, yes, so there are massive cleanup events. You know, we have funding systems through EPA that, you know, these Superfund sites that are the government's paying to clean up the messes that corporations made. I wouldn't be surprised if DuPont and 3M are going to be required to put, it's really hard to, the aquifers that are contaminated with like the PFAS chemicals, it's more likely that they're going to be installing filters in people's houses to take it out of their drinking water than being able to clean the aquifer. And, you know, that might happen in White Bear Lake where their drinking water is contaminated. So, you know, I think, I just saw a thing today, there's like 26,000, I think I maybe said today, 26,000 wells, aquifers contaminated with these Scotchgard-like chemicals.
In Minnesota?
Across the country. Yeah, and they're also, so these PFAS are also in like firefighting foam, so, and so like in a lot of like industrial or even military complexes where they were working with these foams, they've, you know, contaminated. They spray this stuff out on the fields and then it doesn't go anywhere and it just leaches down through and ends up in the water. So, good question.
I know you're not a nutritionist, but I was listening to a podcast which said that, like you mentioned iodine and the effect it has, that the diets and the fads, etc., which go on, some of the nutrients people do not take because they do some of this dieting, might have an effect on gene expressions, etc. And that's completely a different study than the chemicals. But people are saying that that might also cause a lot of these chemical and hormone disruptions. Do you think that might be something going on too?
I do agree, especially as like, you know, we start using like highly concentrated, like, to be honest, like soy proteins and things like that. There are, you know, environmental estrogens, natural plant-like estrogens, and we're ingesting these. You know, a lot of people don't buy iodized salt anymore. That's like one of our main sources of iodine. You know, so yeah, diets is an important player in it, especially like, you know, if you're someone of childbearing age. You know, these are the things that you want to be thinking about if you're thinking about getting pregnant, and make sure that your axis is in place for when you have a fetus. You know, the same hormone you use, the same hormone that's used in a home pregnancy test that's detected in the urine is also signals to that mom's thyroid to increase her production of thyroid gland during pregnancy, right? So it's not just maintaining the placenta, but it's actually signaling to her thyroid to help that output. So yeah, I do think it's something to be thoughtful of. Yeah, Dr. Land.
Well, we have another online question.
Oh, okay.
So Aditya asks, or says, we've seen these effects on mice, but how does this correlate with humans? Most humans are still born without impairments. Has contamination increased since 2000s, or is this the same as before?
So I see two kind of questions in there. So I'll say that there are, whereas some contaminants are decreasing, others are increasing, right? So like, you know, triclosans has been increasing, but since 2016, now it's starting to fall. PCBs historically have been falling since the late 70s when they were banned. But no one's, you know, they're just changing the chemical structures and we're not looking at all the new ones, and those are going to start increasing, right? So, you know, the PFASs were introduced in the, 50s, and then 70s, there were tons of them. What was the first part of that question?
How does it correlate with humans?
How does it correlate? So that's so there are a lot of epidemiologists that do these association studies where they take blood from the population, and also in that blood they look at what chemicals are present, and they try to kind of, you know, make an association between the levels of circulating thyroid hormone and the levels of a chemical, and they do this in a population study, and you can see trends, right? And and there from the 90s, there were these fish eater studies, maybe a little earlier than that, which looked at PCBs for people that were eating fish out of very contaminated places with PCBs, and they bioaccumulate. And these studies were very clear that individuals with the higher levels of PCBs had lower levels of circulating T4. And those same association studies have been done with cord blood. They definitely impact humans, but to really understand if someone has an unknown deficit in hearing, is it related to some chemical exposure earlier in life? You would have had to catch it in that window. So it's hard to answer that.
Any other questions online? Any other? Whoops,
Abby over
oh,
here.
okay. All right. Here I come. Probably this question, maybe one more before you make your way to the stage, President Inch.
Thank you. You did a great job, by the way. It was very clear and great to follow. My question is, do you know if the thyroid hormone functions to affect the middle ear and ossicles in regard to development? So more towards like hypo or hyperthyroidism affecting the middle ear's ability to relay those mechanical sound vibrations from the outer to inner ear at all?
Yeah. So actually that work has been done. It was done when I was a postdoc in the lab that I did my postdoc in at the National Institutes of Health. And Dr. Emily Cordes was the one that kind of looked at that. And it made a lot of sense to actually look at those bones, because we already know that like thyroid hormone controls skeletal growth, right? And she did find that the bones were abnormally shaped. And she used kind of a genetic model of hypothyroidism. She didn't use like the chemical style that we're using here. There's a couple of genetic models of congenital hypothyroidism. She used one of those, and sure enough, the bones were disrupted.
Any other questions? These are just absolutely terrific. Anybody else? Okay.
Thank you.
Looks like one more. One more.
On behalf of all of the humanities majors in the room, Dr. Sharlin, thank you for explaining this in such a clear way. It was just a wonderful presentation, and I'm not going to say easy to understand, but I didn't feel lost.
Thank you. Yeah, I appreciate that a lot, because it's hard sometimes to find the right balance.
Yeah.
So.
Okay, excellent point, Jennifer. You're up.
Well, thank you, Dr. Sharlin, for sharing your research with us. It's really clear to me why the selection committee picked you. It's an important topic, one that I bet most people did not have anywhere near the depth of understanding you do, and thank you.
Yeah, thank you. Hopefully you'll learn this. No problem. Yeah.
You know, I've always been aware how delicate our bodies are and how carefully we are balanced so that we can do things like see and hear and walk and think. I had no idea that the chemicals in our environment can disrupt that balance so profoundly. I think research like this, especially research that engages our students so that they can go on to become doctoral students, they can go on to shape the fields that help us better understand how the chemicals in our environment and the practices that we have to make lives easier sometimes have such profound consequences. And keeping us aware of that and doing research that points out how we are affected is so terribly important. And not just for the purposes of the lecture now and the research that comes of it now. I just think about the legacy that you help create by teaching our students to have a passion for questioning, understanding, developing approaches to answer some of the questions like, what do we do? So I appreciated the lecture greatly. So tonight, we get to conclude this evening by looking at our next opportunity to come together for the Douglas R. Moore Lectureship. And tonight, I'm very proud to announce that next year's lecture award is going to Dr. Maranka Day in the Department of Biological Sciences. So, Dr. Day, would you please stand? I so look forward to hearing your remarks next year, knowing that you're one among several lectureships that have made such a profound difference in the way we think on our campus, the kinds of research we do, the students we produce, and mostly the good work that we're able to do to help inform our communities, our world, and the scholarship around the kinds of important issues that we talk about. So I would like to acknowledge the work of the committee in making its choice for this year and next year. And I would like to commend all of you for coming to listen to such an important and interesting lecture. So thank you all for being here. Thank you for those of you that put this together tonight. Have a great evening. There's a reception outside immediately following. Take care. Safe travels home.